This is a plasmid, a circular piece of bacterial DNA, and it is one of the coolest and versatile things in molecular biology and gene editing. Through years and years of genomic research and countless research endeavors, synthetic biologists have been able to identify all the essential parts of plasmid DNA. So now, thanks to these amazing people, we are indeed quite capable of bending the genetic code itself, transform, modify, and even create the custom sequences which have never before existed. We're living in an era of genomic revolution where the science fiction is now a reality. In order to truly understand how the real synthetic biology works when it comes to plasmid design, we have to study all the different pieces of plasmid DNA. So, let's begin. DNA, deoxyribonucleic acid, is a biopolymer which is mainly comprised of 4 types of nucleotides: adenine, guanine, thymine, and cytosine. Adenine pairs with thymine, and guanine always pairs with cytosine. But we kinda all know that, so let's put it simply. Any biological compound, any sort of organic matter, as long as we know the correlating genetic sequence, can be synthesized. That's the real truth here, no more, no less. So we need a cassette, where a botanist like myself will be able to paste that kind of info and let the nature do the rest. In our case, that cassette is a circular piece of DNA called plasmid. If we will be able to study the components of plasmid DNA in depth, all the small nuances, where the transcript begins, where the insert is, how the resistance sites work, how to figure out if the expression's present, and many, many more, then we will be able to rewrite the code of life as we please. And you can just imagine how cool is that. One of the most important components of your plasmid DNA is your insert itself, a gene of interest that is basically a correlating sequence of a protein you want to synthesize. Say green fluorescent protein or a lax operon for bacterial bioluminescence. So you'll find the correlating sequence using a database like GeneBank. That's basically your insert, but in order to initiate the transcription, you also need a promoter upstream your gene of interest. Promoters basically this piece of DNA which will initiate the transcription, aka DNA to RNA. And after the transcription of our gene of interest complete, we have a stop codon, either TGA or TAA, and then a terminator sequence which will quite literally terminate the transcription. Well, there are other components too. One of the most important one when it comes to bacterial selection is antibiotic resistance site. Let's say your resistance site codes for resistance against kanamycin. When you insert your plasmid into an E. coli, after you make an agar stops, you add that specific antibiotic to your plate. As a result, the only E. coli which will survive will be those who have been genetically engineered to express the resistance against that particular antibiotic. Now that we sort of understand the individual components of plasmid DNA, well, how do we physically cut the DNA to introduce the new genetic material? This, my dear friends, is done by none other than restriction endonucleases, which are enzymes that recognize the specific DNA sequences and cut up DNA on that particular part. And as we know the recognition sites, we can design our insert accordingly, and through DNA cloning technologies like Gibson assembly and Golden Gate cloning, we can actually fuse the insert into a now linear digested DNA, and by the mighty power of DNA ligase that sticks the DNA together again, we can make a circular piece of DNA with our insert now in it. There are numerous other individual parts of plasmid DNA which we will not be able to cover it here, but here's a book which contains it all. It's a great resource from Gene Plasmid repository, and the coolest part, it's entirely free. You might be wondering, now that I know how those components work, how do I actually design a piece of DNA by my own? Well, good news for you. There are numerous online plasmid assembly platforms, but the one I use is BenchLink. It's a free-to-use versatile platform with a clear and easy-to-understand structure. BenchLink's really great. You just create a free account and you're good to go. create all the custom new never before existed monstrosities, new constructs. The only limit is your imagination.